Barnacle Abundance and Diversity within Point Pleasant Park, presented by Lukas Sawatzky. Rocky intertidal zones are dynamic environments where marine and terrestrial environmental aspects create unique ecological gradients. Barnacles are essential crustaceans and key to the inhabitants of this zone. They display distinct patterns of abundance which can be influenced by tidal exposure. Their distribution provides valuable insight into the ecological process of governing intertidal communities, such as stress, predation, and competition for space. Understanding these patterns is crucial for marine biology, particularly in the context of environmental change and coastal management. We respectfully acknowledge that the land and waters where this research was conducted, Point Pleasant Park, is the traditional and unceded territory of the Mi'kmaq people. We honor their enduring connection to this land and recognize the wisdom they bring in understanding ecological relationships. Embracing the concept of two-eyed seeing, which integrates Indigenous and Western scientific knowledge, we aim to enrich our understanding of barnacle ecology by considering both traditional ecological knowledge and contemporary scientific methods. This approach fosters a deep and more inclusive understanding of the natural world and the part we play in. This study investigates the distribution of barnacles across different tidal heights in the Rocky Intertidal Zone at Point Pleasant Park. By examining barnacle abundance from the upper to lower intertidal zones, we seek to determine if tidal height significantly influences their distribution. Our null hypothesis for this study is there is no significant difference in barnacle abundance at different tidal heights along the rocky substrate at Point Pleasant Park, and this research project aims to contribute to the broader understanding of intertidal ecology and supporting conservation and management efforts by highlighting the factors that shape the distribution of key species in the coastal ecosystems, such as barnacles. To identify barnacle abundancy at different tidal heights, we made out 10 zones, each measuring 10 by 10 centimeters across varying tidal heights. The zones were placed to cover 5 different tidal heights: the high intertidal zone, the high mid-intertidal zone, mid-intertidal zone, lower mid-intertidal, and low intertidal zone. These zones were strategically placed to represent each of the tidal heights. Precise locations were marked to ensure consistent data collection throughout the study, and we gathered all the necessary materials, including measuring tapes, waterproof markers, and data sheets, and the camera to facilitate accurate and systematic data collection. During low tide, we collected their barnacle counts in each of the 10 marked zones. Environmental conditions such as temperature and weather were recorded during each visit. There was only one visit, to account for any external factors that might influence barnacle distribution. Consistency in data logging was maintained to reduce variability, and we used our standardized data sheets to ensure efficiency and accurate data entry, enabling us to systematically log and analyze the data collected from the study zones accurately. In these tables, we observed the distribution and averages of the barnacles across the various tidal zones of Point Pleasant Park. Each quadrant within the intertidal area we surveyed, we recorded the number of barnacles present. The highest counts were found in the lower mid-intertidal, and low intertidal zones, with quadrant 9 showing the highest count of 22 barnacles, and quadrant 1 showing the lowest count of 1 barnacle. Now, the table for the averages summarizes the average of barnacles per tidal zone. The highest average barnacle count was observed in the low intertidal zone at 20.5 barnacles, followed by the lower mid-intertidal zone with an average of 15.5 barnacles. These findings suggest a gradient of barnacles abundance from higher to lower tidal heights. This graph illustrates the average number of barnacles observed across the different tidal zones, and the data reveals a clear trend of increasing barnacle abundance as we move from the high intertidal zone to the low intertidal zone. That is, from where there is less water present throughout the day to where there is more water present, and with water that comes waves and nutrients washing on shore. Specifically, the lowest intertidal zone shows the highest average barnacle count at 20.5. The trend suggests that barnacle distribution is influenced by tidal height, with lower zones experiencing higher densities, and this pattern is crucial for understanding the ecological dynamics of the intertidal habitats and can inform conservation and management strategies in coastal ecosystems. Comparing our findings with Moira Flagstan's study on Balanus glandula in Vancouver provides valuable insights into regional variations in barnacle responses to intertidal gradients. While their studies uses basal diameter as a measure, our study at Point Pleasant Park was for the abundance of barnacles closer to the water. Flagstan noted no significant size differences among intertidal zones, potentially due to similar environmental pressures across the study area. Our findings show distinct barnacle abundance patterns across tidal heights, suggesting varying ecological pressure of adaptive responses at Point Pleasant Park. This comparison underscores the importance of local environmental contexts in shaping intertidal community dynamics and calls for further exploration into the regional nuances to enhance coastal management strategies between those of Vancouver and those of Nova Scotia, specifically Point Pleasant Park. Our study shows the intricate biological and ecological dynamics that govern barnacle distribution across different tidal heights. Barnacles in intertidal zones are influenced by factors such as competition for space, predation, pressure from other species, and substrate type, and the exposure to wave action and sunlight. Our observations of higher barnacle densities in lower mid-intertidal zones align with theories of optimal environmental conditions and competitive exclusion in this habitat. These findings resonate with the studies of Moira Flagstand, which emphasize the role of biotic and abiotic factors in structuring intertidal communities. Understanding these dynamics not only enriches our ecological understanding, but also informs conservation strategies aimed at preserving biodiversity in coastal areas. While our study provides valuable insights, it's necessary to acknowledge its limitations. Variability, given weather and temperature conditions and fluctuations, may have influenced our sampling, potentially affecting barnacle counts. Additionally, our study's spatial scale and sample size could be expanded to enhance robustness and generalize findings across broader geographical contexts. It is also important to note that Point Pleasant Park is only one intertidal zone and coastal area of Nova Scotia, whereas there are several with different barnacle species populations which could be further examined. Future research should consider longitudinal studies to capture temporal changes in barnacle distributions and integrate indigenous knowledge through collaborative approaches, aligning with the principles of two-eyed seeing. This inclusive approach not only enriches specific inquiries, but also fosters sustainable management practices that respect and integrate traditional ecological knowledge, where we can work together to both preserve and examine the ecosystem. Moving forward, efforts should focus on adaptive management strategies that promote resilience and conservation of intertidal ecosystems in the face of global environmental changes. To wrap up our presentation, our study demonstrates significant variations in barnacle abundances across tidal heights, challenging the null hypothesis of no difference. The lower intertidal zones support higher barnacle densities, possibly due to more stable environmental conditions and greater access to resources and nutrients through wave contact. These findings underscore the ecological importance of understanding our local intertidal dynamics for effective coastal management. Moving forward, integrating principles of two-eyed seeing can enhance our understanding by bringing Western science approaches with indigenous knowledge systems, promoting holistic conservation strategies. Future research should explore broader spatial and temporal scales to capture seasonal variations and long-term trends in intertidal communities. By doing so, we can better inform sustainable management practices and safeguard these critical coastal ecosystems. In these acknowledgements, I would like to express my sincere gratitude to Isabelle Ube for her invaluable guidance and support throughout this research process, and a thank you to the Dalhousie University for providing the opportunity to conduct this study as part of the Bio 3664 Intertidal Ecology class. I'm also grateful to the management staff at Point Pleasant Park for their cooperation and support during the fieldwork and in their management of the property. Additionally, heartfelt thanks to my friends for their dedicated assistance with data collection and for making this research experience both productive and very enjoyable. Fieldwork is a wonderful thing, and it is even more wonderful with friends.